You measured the flour down to the gram, you checked the salt, you followed the recipe exactly, and 3 hours later, you opened the bowl to find a flat, dense, sulking lump of dough. The mistake usually happens in the first 30 seconds because of a basic misunderstanding of what is actually in the bowl. Yeast is not a dry baking ingredient. It is a living colony of single-celled fungi called Saccharomyces cerevisiae. These organisms eat, breathe, and reproduce, and they are exquisitely sensitive to their environment. The temperature of your kitchen, your flour, and your water dictates whether this colony thrives and produces carbon dioxide, stalls out entirely, or dies on contact. To control that environment, you need a single piece of equipment, a $10 instant read thermometer. Track the temperature of your dough instead of relying on the clock, and you stop guessing. We will look at 4 exact thermal milestones that determine whether your bread rises or collapses. Let's start with hydration. For standard active fry yeast, the kind in the paper packets, the cells need a specific thermal window to rehydrate and wake up. That window is precisely 100 to 110 degrees Fahrenheit. Most recipes call for warm water. The problem is that human body temperature is around 98 degrees, so when you turn the tap until the water feels noticeably warm to your hand, you are usually pulling water between 120 and 130 degrees. This animated thermometer scale shows us exactly where that leads. Past the yellow zone, we hit a hard red line at 140 degrees Fahrenheit. At this specific temperature, cellular proteins denature. The enzymes unfold. The entire yeast colony collapses and dies instantly. You might think the safe route is to use cold water and just let the dough rise longer. But dropping yeast into water below the mid-60s triggers a different mechanical failure. When cold shocked, yeast cell walls become abnormally permeable. In this diagram, an amino acid called glutathione leaks out of the cell into the dough. Glutathione acts as a powerful reducing agent. It actively attacks and breaks apart the disulfide bonds holding your gluten network together. Your dough becomes slack, sticky, and unable to hold its shape. So missing your water temperature on either the hot or cold side destroys the structural integrity of your loaf before you even start mixing. Once your water is correctly measured, your target for the mixed dough itself is 78 degrees Fahrenheit. Professional bakers track this number religiously because it balances 2 competing organisms. 78 degrees is warm enough for the yeast to produce carbon dioxide at a steady rate, but cool enough that the lactic acid bacteria living alongside it can still thrive and develop complex flavors. Hitting that 78-degree mark requires factoring in a hidden variable most home bakers miss entirely: the physical work of kneading. This equation breaks down how dough absorbs ambient heat. If you run a stand mixer for 10 minutes, the friction of the dough hooks stretching the mass converts mechanical energy into thermal energy, driving the internal temperature up by 10 to 15 degrees. If you started with room temperature flour and warm water, that extra friction pushes your dough past 90 degrees. The yeast shifts into thermal overdrive, producing carbon dioxide so fast that your 2-hour fermentation window shrinks to a single hour. Your stand mixer is an active heating element. Compensating for that friction, usually by starting with cooler water, keeps the fermentation schedule intact. When your dough is resting at the right temperature, the yeast eats sugars and expels gas. The bread dough traps that gas using gluten, which acts as a microscopic suit of chainmail made of hydrated proteins. Here, the chainmail network expands. As long as the protein strands stretch faster than the gas produces, the dough rises. But if it ferments too long, gas outpaces elasticity. The walls thin, merge, and the protein matrix violently tears. Gas escapes, leaving a collapsed loaf. To prevent this, you can test the physical tension of the dough using a 10-second diagnostic check known as the poke test. Press your fingertip half an inch into the dough. If the indentation springs back immediately, the gluten is still tight and needs more time. If it springs back slowly and leaves a shallow dent, it is ready to bake. If the dent stays flat and the dough sighs, the structure has failed. You can manipulate this timing by placing the dough in the refrigerator overnight. At 38 degrees, yeast slows to a crawl, holding the structure intact. But the lactic acid bacteria keep working, generating deep, complex flavors over 12 to 24 hours. Relying on a recipe's exact timing for this step guarantees failure. A massive, fully risen dough looks like a success, right up until the moment it structurally collapses, so you have to test the tension directly. The final test happens in the oven. When you slide your dough into 450 degrees of heat, the yeast does not die immediately. It accelerates. As the internal temperature climbs from 78 to 120 degrees, the yeast metabolism goes into overdrive. This final, explosive burst of gas production is called oven spring, giving the loaf its last surge of volume. This acceleration is brief. As the internal core hits 120, the cells weaken. At 130, they are dying, and by 140 degrees, the entire colony is dead, and the gluten and starches set into their permanent shape. We can trace the entire lifespan of the bread through these 4 markers: water measured to activate the cells, a dough target of 78 degrees, a test to confirm structural tension, a final cap at 140. Bread is a precise biological system. Control the thermal environment of the colony, and the results take care of themselves.